Dear students, today let us learn about the concept of plant biotechnology. Plant biotechnology is the process to produce genetically modified plants by removing genetic information from an organism, manipulating it in the laboratory, and then transferring it into a host plant to modify certain of its characteristics for the benefit of mankind. Two main techniques of plant biotechnology are plant tissue culture and plant genetic engineering or plant transformation. Let us now start with the concept of plant tissue culture. Plant tissue culture is also referred to as in vitro, xenic, sterile or cell culture. It is the aseptic culture of plant cells, tissues, organs and their components under defined physical and chemical conditions in vitro on an artificial medium containing all the nutrients required for the normal growth and development of plants. Then let us see the basic concept of plant tissue culture. The technique of plant tissue culture depends mainly on three basic concepts. The first concept is totipotency. It is the potential or inherent capacity of a plant cell to develop into an entire plant in response to suitable stimuli. The next concept is de-differentiation. De-differentiation is the capacity of mature cells to return to meristematic condition and develop a new growing point, followed by redifferentiation, which is the ability to reorganize into new organ or plantlet. And the third concept is competency. Competency is the endogenous potential of a given cell or tissue to develop in a particular developmental pathway. Let us now see a brief overview of plant tissue culture process. Plant tissue culture technique has led to the increase of knowledge in the following fields. The first is the totipotency, nutrition, metabolism, division, differentiation and preservation of plant cells. Next is morphogenesis and regeneration of plant from individual cells or tissues through organogenesis or somatic embryogenesis. The third is somaclonal variations or variations generated through in vitro culture. Next is in the production of haploids through anther and pollen culture or ovule culture. The fifth is in vitro hybridization through ovule, ovary and embryo cultures to overcome both pre-zygotic and post-zygotic sterility barriers. The sixth is micropropagation or clonal propagation of plant materials. Next, in vitro mutagenesis studies for selection of mutants tolerant to biotic and abiotic stresses. The eighth is in vitro secondary metabolite production by cultures. And finally, in vitro regeneration of genetically engineered plants. Next, let us see the concept of plant genetic engineering or plant transformation. Plant genetic engineering or plant transformation is the method of integrating genes with desirable traits into host plants and recovery of transgenic plants by plant cell and tissue culture technique. Let us see the basic steps of plant genetic engineering now. The basic steps of a general plant genetic engineering process includes: the first step is selection of a plant gene whose introduction in other plants would be of interest. The second step is identification and isolation of the target gene of interest. The third is transference of isolated gene to the host plant cell. And the last step is the regeneration of complete plants from transformed cells or tissues. Now coming to the tools of plant genetic engineering. Plant genetic engineering uses key tools like restriction enzymes, polymerase enzymes, ligases, vectors, and the host organism, and genetic transformation in plants can be achieved most commonly by the following gene transfer techniques. The first common gene transfer technique used in plant genetic engineering is the indirect gene transfer or vector-mediated method. The next common method for gene transfer in plant transformation is the direct gene transfer method. One example is the agrobacterium-mediated gene transfer method. In this method, the target DNA for the desired trait is isolated from the original organism and inserted into agrobacterium, which is used to infect the target plant cells. The transformed plant cells containing the target DNA are then regenerated in vitro into plants with a new trait. The next common method for gene transfer in plant transformation is the direct gene transfer method. One example of such method is the biolistics or gene gun method. In this method, DNA that codes for the desired trait is coated onto tiny particles of tungsten and fired into a group of plant cells with high velocity. Cells that accept the DNA are grown into plants with the desired trait. Let us see the goals of plant genetic engineering. Plants are genetically engineered to modify certain of its characteristics and produce transgenic plants with improved traits like: first one is plants that are resistant to herbicide, insect, fungal and viral pathogens. And the next one is plants with improved protein quality and amino acid composition. The third modification is for improvement of photosynthetic efficiency in plants. And finally, for production of plants with improved post-harvest handling. Let us now see the history of plant tissue culture. The science of plant tissue culture takes its roots from the discovery of cell, followed by propounding of cell theory by Sladen and Swan in 1838. They visualized that cell is capable of autonomy and therefore it should be possible for each cell to regenerate into whole plant if given the suitable environment. Based on this premise, Gottlieb Haberland in 1902 developed the concept of in vitro cell culture and predicted totipotency. He was also the first to culture isolated single fully differentiated individual plant cells in Knopf's salt solution enriched with glucose. He observed in his cultures that the cells increased in size and accumulated starch but failed to divide. Despite the lack of success, Haviland made several predictions about the requirements in media in experimental conditions which could possibly induce cell division, proliferation and embryo induction. Therefore, Haviland is regarded as the father of plant tissue culture. Then, learning from Haviland's failure, Henning in 1904 excised nearly mature embryos from seeds of several species of crucifers and successfully grew them to maturity on mineral salts and sugar solution. The next most successful of the early attempts in plant tissue culture involved development of callus tissues from cultures of maize, pea and cotton root tips reported independently by Cote and Robbins in 1922. Following this, White in 1934 reported that not only could cultured tomato root tips grow but could be repeatedly subcultured to fresh medium of inorganic salts supplemented with yeast extract and 2% sucrose. Later, in 1937, he replaced yeast extract by vitamin B, namely pyridoxine and thiamine, and proved their growth-promoting effect. Again, in 1939, the possibility of cultivating plant tissues for an unlimited period was independently endorsed by Gottfried, White and Novkov. Adding to the ongoing improvements in the culture media, providing tremendous impetus for further work in embryo culture, Johannes van Overbeek in 1941 reported growth of seedlings from heart-shaped embryos by enriching culture media with coconut milk besides the usual salts, vitamins and other nutrients. Following this, Michael Levin in 1947 reported the spontaneous formation of shoots and roots on cultured carrot tissue, and these observations turned attention to the question of totipotency. Meanwhile, Skoog and Sui in 1948 demonstrated that the amount of shoot and root formation depended on the proportions of auxin and adenine supplied in the culture medium. Further, Skoog and Miller in 1957 demonstrated that tobacco callus tissues retain the potentialities of the zygote to form both shoots and roots by adjusting the relative concentrations of auxin and kinetin in the culture medium. And in 1962, Murasige and Skoog prepared a medium by increasing the concentration of salts 25 times higher than Knopf's salt solution, and it enhanced the growth of tobacco tissues by 5 times. Till today, Murasige and Skoog medium or MS medium has immense application in tissue culture. Despite continued efforts by Haberland's collaborators and others, no significant progress on cultures derived from single cells was made until Muir and his co-workers in 1958 demonstrated that when callous tissues were transferred to liquid medium and subjected to shaking, callous tissues broke into single cells. And further, the plating technique for cloning of the single cells by filtering suspension cultures was developed by Bergman in 1960. Then, in 1958, Stewart and his co-workers observed the phenomenon of somatic embryogenesis in suspension culture of carrot, thereby proving cellular totipotency and validating the ideas of Haberland. In 1962, another milestone was achieved when Kanta and Maheshwari developed a technique of test tube fertilization. Further, the objective of development of whole plant from the proliferated tissue of cells were first achieved by Wassel and Hildebrand in 1965 by regenerating plantlets from colonies of isolated hybrid cells after cross between Nicotiana glutinosa and Nicotiana tabacum. Meanwhile, in 1966, Guha and Maheshwari cultured anthers of datura and raised embryos which developed into haploid plants initiating androgenesis. This discovery received significant attention since plants recovered from double haploid cells are homozygous and express all recessive genes, thus making them ideal for pure breeding lines. Next breakthrough in application of tissue culture came with isolation and regeneration of protoplasts, first demonstrated by Cocking in 1960. Then, in 1972, Carlson isolated protoplasts from Nicotiana glauca and Nicotiana langsdorffii and fused them to produce the first somatic hybrid. Thus, the early research in plant tissue culture demonstrated that tissues isolated from plants can be grown in culture for indefinite periods of time. They can produce shoots and roots, and finally, single isolated cells in culture can produce embryos. Let us now see the history of plant genetic engineering. With the discovery of restriction enzymes, reverse transcriptase, and the development of techniques to produce recombinant DNA in vitro in the early 1970s, tissue culture headed towards a new research area. The totipotent plant cells could now be altered by insertion of specific foreign genes to produce genetically modified crops by using the bacterium Agrobacterium tumefaciens. In 1974, Zenon and his co-workers discovered that TI plasmid is the tumor-inducing principle of Agrobacterium in crown gall disease in plants. This was followed by its successful integration in plants by Kilton and his collaborators in 1977. And in 1980, Zambrowski et al. Isolated and studied the detailed structure of tDNA and its border sequences. Soon thereafter, in 1984, transformation of tobacco with agrobacterium was accomplished to develop transgenic plants. Simultaneously, there was an upsurge in development of techniques for large-scale DNA sequencing, and this was followed by complete genome sequencing of rice in 2005. Thus, laying foundation of genomics, which is the main focus of present-day plant biotechnology. Coming to the conclusion, plant biotechnology, including aseptic tissue or cell culture and the genetic modification of plants, resulted from basic research findings and technological innovations. The early research in plant tissue culture demonstrated that isolated plant tissues from plants could be grown in culture for indefinite periods of time and could be induced to produce shoots and roots. And finally, single isolated cells in culture can produce embryos. These studies provided the platform for genetic transformation of plants. Thus, plant cell culture has made great advances in association with plant genetic engineering, and dramatic progress has been made in the last few years in the development of gene transfer system for higher plants.